There are numerous ways to describe a biological pathway, and for many, a diagram is as good a way as any. A diagram acts as a visual representation of events, and when well constructed, they should be intuitive and easy to understand. The problem is that there are so many ways one can draw a diagram. There are also few rules about how to represent the various types of interactions that can occur between components of a biological system. The result is lots of different pictures, but together they are not a coherent resource that can be collated and understood as a whole. In the accompanying PLoS biology paper, we describe a pathway drawing system that allows one not only to represent a complex range of biological events in a logical and standard manner, but also to use the diagram to run simulations of the pathway's activity. It begins with a pathway notation system called MEPM, which allows one to construct diagrams that document the known events of a pathway in a graphical form. In this system, each step in a pathway is explicitly shown if known, and a complex series of events can be depicted in an unambiguous manner. Once complete, the diagram represents what you think you understand about a system. This is all good and well, but how might this pathway actually work? To explore this question, we have built a computational modelling system that allows you to simulate and visualize a pathway's activity. This is based on something called stochastic Petri nets. Petri nets are networks made with two types of node, called either places or transitions. In this notation system, these concepts map onto the idea of pathways being composed of biological components and processes. The first step in performing a simulation is that you need to parameterize your model. To do this, you simply place a transition node at the different inputs into the pathway model. Then you need to place tokens on these nodes. Tokens represent the initial amount and/or the activity of a pathway component at the start of a simulation. You might know something about this from experimental data. Tokens move from one component to another when a transition node is fired. The transmission of tokens through a transition node is based upon certain simple rules and is essentially random, or in other words, stochastic. During a simulation, tokens will move down the pathway and represent the activity status of a given component at a fixed point in time. When you load a pathway into a tool called BioLayout Express, it will ask you, would you like to run a simulation? On answering yes, you can set the parameters for the simulation, in particular the number of runs and time blocks. The more runs you perform, the less variable the answer, and the number of time blocks control the length of the simulation. Calculation of the results of a simulation usually takes only seconds, even for relatively big pathway models. You are now ready to examine the results of a simulation. You can place your cursor over a given component and it will reveal a graph of its activity during the simulation. Alternatively, you can select a number of nodes and view their activity and name within the class viewer. Finally, and most importantly, you can visualize flow using an advanced visualization engine. Once you get the hang of this modelling system, there are a few limits to the size and complexity of the models you can create. This is a model of the influenza A life cycle within a human macrophage. It was created over a 4-month period by a postdoctoral biologist with no experience of modelling prior to its assembly. She read the literature, examined other diagrams, and gradually encapsulated this knowledge into the model. She then parameterized it based on known timings of events and what other facts about the dynamics of infection she could glean from the literature. Initially, she focused on the virus life cycle and then built into it the host defense systems known to be present within a macrophage. We have been using this model to better understand the complex interplay between host and pathogen, and it has been helping us better design our experiments. Models such as these allow you to think differently, to think logically and at a systems level about the biological pathways important to you. They allow you to better understand what is known, and almost as important, what is not known about any given system of interest. If you build a model yourself, it reflects your knowledge and interest, not somebody else's. You can use models to easily make virtual gene knockouts and to simulate the effect of such a change to the activity of the pathway. Or perhaps allow you to test the effect of changing the initial conditions, such as priming or in a virtual macrophage with interferon prior to infection. Whatever your pathway of interest, we believe the modelling system described here can help you better understand it. Metabolic pathways, signalling pathways, or interactions between different cells and tissues. Or perhaps even a way of documenting what proteins are associated with a given complex or structure. Once created, models can be edited and shared with others. They can be used to help interpret the results of experiments or help design your next piece of research. They can also be used for teaching, or as in our case, used to get students to demonstrate what they understand through model construction. So whatever your interests, we hope that the pathway modelling system described here is of use to you.